Okay, this is podcast number 2 for Standard 3.5. In this one, we're going to take a look at how Gregor Mendel, our father of genetics, did some experiments with pea plants so that he could come up with his law of segregation. So when Mendel was working with his pea plants, remember that he had noticed that there are 7 different traits within pea plants, and each of those traits had 2 different forms. And remember, we call those different forms. So these 7 traits that he saw were actually genes that he was looking at. And we call those different forms of the genes that he saw. Each one had 2 different forms. Those are the alleles. So he started to get curious about these different forms of these genes that he was looking at, and he started to do some experiments with them. So the first thing he had to do is he had to identify some true breeding plants. True breeding, you can also use the word pure. If you're talking about true breeding, it's also the same as homozygous, which we learned in the last podcast. Okay, so in a true breeding plant, these are when the parents have the same traits and they're going to produce offspring with the same traits. So every time that he took 2 plants and bred them together, those 2 parents had the same traits and all of the offspring ended up having the same traits as well. That could happen in the case of the pea plants because there was only 7 traits that he was looking at. And he was really smart about it, and he only looked at 1 trait at a time, and he made sure that 1 trait was what was being shown all the time. So basically what he would find is he would take 2 tall pea plants. So he'd have 1 tall pea plant and breed it with another, and every time that he did that breeding, the offspring came out tall. Once he identified plants that did this, and it would be the same if he took a purple pea plant and a purple pea plant, and they always made purple, he then identified those as true breeding plants. And he used these true breeding plants in a series of experiments that he did over a number of years on these pea plants to come up with some of his ideas about genetics. So once he had identified his true breeding plants, the ones that he knew were pure, he then did a process called cross-fertilization. Okay, so he took those 2 true breeding plants and he took ones that had opposite forms of the gene and he crossed them. So he made them basically mix their DNA, mix their gametes. So he would take a true breeding tall and a true breeding short, and they would breed. Same thing with a purple and a white, or a yellow and a green. So depending on what trait he wanted to look at that time, he made sure the 2 parents he was going to breed with each other were true breeding. So before, they always had made offspring that were like them. Now he was going to try to mix them, and he wanted to see what would happen if he did that. So he called this first generation the P generation. The P, you can think of, stands for parents. Okay? And these are the first 2 individuals to mate in a genetic cross. And he was actually going to do this over a couple of generations to kind of figure out what was going on. So he had his P generation. There was 2 true breeding individuals. And what he got from that, he called the F1 generation. It actually stands for filial. But the F1 generation are the offspring that got made from this P generation up here. So he took his 2 true breeding parents, bred them together, they had offspring, and he called those offspring the F1 generation. So this is how he did his experiment. In order to be very controlled about it, because the way that flowers are pollinated in the wild, it's not very controlled. It's bees and other insects or the wind that's doing this. So he actually went over and he removed the male parts from one flower, and then, like using a paintbrush, he actually transferred the pollen over to the other part on another plant. So he was actually like making sure that the pollen that he wanted from one plant was going on to the next plant. And then once that pollination happened, then he would get his peas that he would make, and he would plant those peas and he would see what they formed. So you can see why this took many, many years to make this happen, because pea plants, it takes several weeks to grow some pea plants. And you have to have the right conditions, so the weather has to be right. It's not always nice in Austria. Okay, so he took the sperm, right, the pollen from one plant and put it onto the other plant, removing the male parts from that plant just to make sure. Got the seeds, planted them, and then he could see what the offspring were going to look like. Okay, so he did this for all these different traits, and what he ended up finding is that anytime that he took 2 true breeding parents, remember, they were always pretty tall, they always pretty short, crossed them with each other, they always ended up showing only one trait of one of the parents. So they had the trait of one of the parents, not both, and not a mix of the both, but one, they looked like one of the parents. Okay, so for example, in his P generation, when he crossed a true breeding tall with a true breeding short, all of the offspring in the F1 generation, all of their offspring up here, ended up being tall. And he was curious about that because he was like, well, what happened to the short? Okay, he's like, oh, well, that's kind of weird. What we've learned is that sometimes we have alleles that are dominant to others. So we now know that tall is dominant to short. So if the tall allele is there, then the plant is going to be tall. But he didn't really get that at that time, and he did this experiment with all of these different traits, and every single time the same type of thing happened, where he would end up crossing a dominant with a recessive, and they would always end up looking like the dominant. So he was really curious about why that would be. And he wanted to know if the recessive trait had now just been disappeared. Like, is it just gone for good? Or is it going to come back? And so he continued with his experiment, and he did another cross. So we call this a monohybrid cross, this work that he's doing. And remember, mono means one. Okay, so when we do a monohybrid cross, we're doing a cross involving just one trait at a time, and we will do ones with two traits at a time later on. Right now, we're just looking at one trait at a time. Okay, so what he would do is he would make sure that his pea plants were identical in every way but one trait, and make sure that only the one trait he was looking at was different between them. So when you do this in a monohybrid cross, it allows you to just look at that one trait without worrying about what the other traits are doing. So he would take a tall plant and a short plant, and that would be the only difference between them. So their flower color and shape and pea pod shape and color, all of that stuff was the same between them except for their height. Okay, so this is what the important part for a monohybrid cross is. So when he does a monohybrid cross, he is actually going to generate another generation of pea plants. Okay, so what he did is he took his F1 plants and he let them fertilize each other. So remember, he had two true breeding parents. So he had a big T parent, he crossed it with a little t parent, that's the tall and the short, right? And they made some offspring. He let these offspring, that's the F1 offspring, he let them just kind of fertilize each other, okay? And what he found is that in that F2 generation, both forms of the trait ended up showing up again. So when he took those two tall hybrids, right, so that's what you got from crossing the tall and the short, you got your two tall hybrids. In the F2 generation, for every 4 offspring, 3 of them ended up being tall and only 1 of them ended up being short. But the short came back. Okay, so he crossed a tall and a short first, and they all came out tall. He let those guys fertilize each other, and the short came back. So this is where he started to get kind of curious about why would that happen. Okay, and basically what his conclusion to this was that when you cross 2 hybrid tall plants, you get some tall and some short. And he called these plants hybrid because they were a mix of a parent that was tall and short. And so anytime you have 2 different alleles, you know that you have a hybrid. The other word for hybrid is heterozygous. Okay, so here's what this would look like. So here he had his tall plant and his short plant. And remember, the only thing that's different between them is their height. Everything else is the same. They made their F1 generation and they were all tall. Okay, so you could look at it this way if you wanted to put the alleles in here. They're big T, and little t. And you crossed your F1 generation, each of these guys would then end up being big T, little t. Okay, and we're going to talk about why that happens. And then over here, you end up with organisms. 3 of them ended up tall, and 1 of them ended up short. And that is going to happen because of the way that the alleles are going to separate from each other when we make gametes. And he did this experiment over and over again, and he saw the same result happening over and over again. So he crossed purple and white, got his purple hybrids in the middle, let them self-pollinate, got the 3-to-1 ratio. Same thing happened with the round and wrinkle. Always got this 3-to-1 ratio. 3 were dominant, 1 was recessive. Okay? What he has figured out, and what we now know from what we've learned since Mendel's time, is that our genes are actually located on chromosomes. And remember, those chromosomes are homologous, so they're matching pairs. So these two chromosomes are homologous to each other. You can tell they're the same size. And they both have a place on them, we call it a locus, for the flower color gene in this example. This one, this chromosome up here, has a purple allele, and this one has the white allele. Okay? And so if this was in one organism, and you were going to pass on the trait, you would pass on only one or the other. And what that means, and what Mendel ended up calling this, was his law of segregation. So to segregate something means to separate them. And in this case, we're going to be separating homologous chromosomes. Okay? So our homologous chromosomes, remember, we know this already because we talked about meiosis. Homologous chromosomes separate from each other during that process of meiosis. They pair up. They might do a little crossing over. But eventually, they're going to end up being separated from each other. That's how you get those haploid cells at the end of meiosis. Okay, so remember, each gamete that is made during meiosis is going to get one homologous chromosome from the pair. Okay, so remember, all of our chromosomes are paired. They're all nice and paired up, same size. But when you go to make your gametes, they get separated from each other. Right, and they each become going into their own individual cell. Okay, so in humans, we would be talking about 23 sets that would be doing this. Okay. Right, so that means, and you can see this in this picture, this means that each gamete gets one copy of each gene. And remember, we call those different versions of the gene alleles. Okay, so just remember, genes are found on chromosomes. Okay, so when you separate your chromosomes, you're also thereby separating the different kinds of genes that you might have. And so when you go to pass on those genes, you're only giving one copy of every type of gene that you have. Okay. So this is something that you can look at for the law of segregation. So here we're starting with our nice little cell over here. Remember, this cell is now diploid, and it's going to go through its first division, and we end up with our two haploid cells. And at the end of that, each of these cells now has only one chromosome, one version of each type of chromosome in them. So they have one version of each type of gene. So this one had two big G's in it, right? Now it's only got one big G, and this one's got one big G. And this one's got one little g, and this one's got one little g. So you have taken those different genes where you originally had all of these different copies of them, and now you've got one in each cell. Okay, here's another way that you can look at that. So you have a big S, little s. Okay, the gametes get made, so this guy can only give a big S, because that's what he has, and this one can only give a little one. And when fertilization happens here, that big S and that little s are going to come together, okay? And that's going to make your offspring. And the gametes that those ones could make, then this guy could give either a big S or a little s. So we're going to practice with this some in class and seeing how the law of segregation works. And we're going to then apply that to Punnett squares. Okay. One more picture of this. So after fertilization, the homologous chromosomes are going to pair up again. So once that segregation has happened and then you'd fertilize, then each allele will find its partner again. Okay, so one more example here. You have here's just a random thing, purple grain and white grain. Okay, so big G is the dominant, little g, right? You're crossing those two. So this parent right here can only give a big G. So that's what this gamete is showing. This parent over here can only give a little g. That's what this one's showing. So during fertilization, at this point, this offspring becomes big G, little g. So when these two came together, they come up and they find their partner, their allele partner. And that's one of the things that like a lot of organisms can kind of control, is that if all the alleles don't find their partner, that might tell the cell there's something wrong, and it may not fully develop past the zygote stage because of that. Okay, so now that you have this new organism, now if this organism was going to reproduce, it could give either that big G or it could give this little g. And that's what the law of segregation tells us. It's not going to give both of those, it's only going to give one or the other. And we're going to, like I said, use this principle then to move into looking at Punnett squares and how we can figure out basic monohybrid crosses.